the Expert Webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Again, thank you for joining us today. It is now my pleasure to introduce our speaker, Millie Ula from Repligen.
Good afternoon, everybody, or good morning, depending on where you are. I'm very happy to be here today. So I'm going to speak for the next 15 minutes or so on the solutions for process intensification. And this is using a high-performance cell retention device for cell culture process intensification and also continuous process that many of you may be familiar with. So to begin with, I'd just like to review how the market is growing in terms of mAb-based biologics. There are more and more drugs entering the market and 8 in FDA approvals currently. What we see with these new drugs coming onto the market are emerging trends and new challenges. And so these are related to several aspects, including the use of integrated continuous processing, shift to disposables, regionalization, and focus on process economics. And all of these things are really changing the way that we manufacture drugs now and looking forward into the future. If you look at historical products that were manufactured by continuous processes, by perfusion, you can see that it's really not a new technology. The first approved drug to be launched using a perfusion device was a spin filter, and it's ReoPro in 1994. And more recently, what I'm really going to focus on today is the ATF system. You can see we're also in approved manufacturing processes since 2000. So not only is the ATF used for continuous processing, but you can also use it to improve process efficiency in very traditional and classic fed batch. And there are a number of ways that that can be accomplished. So this can be through high-density seed transfers, or through cell banking, or through N-1 perfusion. So you can see up here that we have a high-density cell banking application, and this is the creation of a larger volume frozen cell bank. But then the implementation of that cell bank into a process really allows you to skip a number of steps. Down here, you can see N-1 perfusion. So this is really eliminating either N-1, which is 1 step before your manufacturing reactor, or 2 steps before your manufacturing process would be N-2, and that would be increasing efficiency and reducing time for processing in the N-1 process. And both of these applications can be applied to very classical fed batch applications. So cell retention devices are not only used for continuous, can be used in very traditional processes. So if you're not familiar with the ATF, it stands for Alternating Tangential Flow Filtration. It's essentially a perfusion device, and it is used in GMP manufacturing, stainless steel housing with a hollow fiber filter cartridge inside. It's attached to the bioreactor via a single connection, and the real benefit of this design is that you have a constant flow up through your single connection into the bioreactor and back down again, which with each short cycle you have a small backflush, which really helps to prevent the filter clogging and can accomplish cell concentrations of over 100 million cells per mil or even higher. So I'm going to review a case study now. This is using a perfusion applied to N-1 seed expansion. And this would be something you could apply to a fed batch process. And ultimately what we'll show is that you can reduce the time to manufacturing, because the current status quo of a fed batch could be a process from vial to the manufacturing reactor and to harvest of 30 days or longer. The ATF retention system can intensify that process First of all, apply high-density cryo-seed intermediates, and this is where cell banking comes in, or you could incorporate a pilot-scale ATF perfusion bioreactor in an N-1 step, or you could actually apply both. So in this example, we're going to review both. This slide here reviews the production of the cryoseeds, the cell banking. So the first graph that you see here is showing the actual perfusion process that was used to create the cryoseed. This graph here then shows the thawing of 100 ml seed in a perfusion process, and this one here compares a 1 liter cryo bag with a 100 ml cryo bag. And what you're seeing here is that ultimately the viable cell densities and the viabilities of the cell line from when it was initially manufactured compared to when it was thawed in both volumes is very reproducible and maintains viability of the cell line. In this slide here, what we're reviewing here is the use of that cryoseed and the implementation of that. And finally, when you put that into the production fed batch, how does that affect the process? So you can inoculate at 2 different cell densities, and this graph here shows 3 colors. So the green color is inoculating the manufacturing reactor at 10 million cells, and the blue and the red line here is inoculating the final production reactor at 0.5 million cells. So you can see that by increasing the cell density of your manufacturing reactor, it saves you time. So it's reduced from 14 days to 9 days. And it also increases the cell densities as well, so of the entire batch. So what you're seeing here is the actual production of protein from that same reactor. And you can see very comparable protein production here when inoculated at 0.5 million cells. And when you inoculate at 10 million cells, again, you're really getting a much higher protein production in a shorter amount of time. And ultimately, when you compare the specific productivity of those 2 processes, you can see that the specific productivity of both seeding densities is again the same. So you haven't affected either one, but actually you've achieved the same cell densities and the same product concentrations. This is applied to a very traditional fed batch. So what we've demonstrated in this case study is that the ATF offers significant and production duration. You can do that in a couple of ways, in producing high-density cryo-seed intermediates, N-1 seed expansion, and you can reduce your processing time anywhere from over 30 days, which is very traditional, to less than 14, very comparable cell densities and protein. So if you think about fed batch in relation to perfusion culture or in relation to what we think of as continuous processing, fed batch processes typically have much larger production reactors. In this example, we're showing 5,000 liters here in this picture, but in fact, a fed batch process can have reactors up to 30,000 liters. And this obviously requires more time to set up a new facility, additional steps. There could be additional process risk just because of that much larger volume, and of course additional capital risk as well. The reason why more and more people are looking at continuous processing and perfusions, particularly in early clinical phases, is you have a much better utilization of reactor time. You produce the same amount of product typically in a short time, and the product produced tends to have a more consistent quality through a steady state operation. I'm going to talk more about that. Perfusion in later clinical phases and in scale-up is also lower risk because the development scale tends to be commercial scale. And that's where it really lends itself to single use. So one of the reasons why people hesitate to change to processes is the understanding of how the FDA defines a batch. And there seems to be a lot of history around this idea where one bioreactor volume is thought to be the definition of a batch, and this lends itself to what we've typically been doing historically in fed batch operations. Actually, the FDA presented this information just a couple of years ago where they stated that a batch is a specific quantity of a drug or other material that's intended to have uniform character and quality. So it's not just one bioreactor volume. It can be applied to the quantity of material and not necessarily specify the mode of manufacture of. So perfusion actually enables more consistent product quality. So here you see a graph with a typical fed batch growth profile, and there are two runs shown here. And then you have actual data showing perfusion and steady state. So what we're showing here is really in a fed batch process, you have continuous growth, logarithmic growth, and then you reach a peak, and then there's time. So there's only really 2 points during this process where you have very similar bioreactor characteristics and cell concentration characteristics compared to the perfusion system here where you have steady state. And the idea here is that during this steady state, your metabolism of the cell is very consistent, so your production of your protein is also very consistent, and that allows you to have many benefits in terms of consistency. Here we're looking at how we could potentially define a batch. So typically, we've historically defined a batch by the bioreactor volume following the normal life cycle of the cells. In a continuous process, you can apply a batch in a number of different ways. So here you can see potential to pool during a steady state, and this would require every 3 days pooling of the harvest, and then that would have several benefits because it could be used to help to manage downstream operations, or it could be 1,000 liters of perfused harvest pooled volume, or it could be the actual protein concentration volume. So there are several ways that a process could be defined in a continuous. Very much depends on the process how that could be defined, but it's pretty flexible to be able to define a batch. That automation, just like any type of system, also helps to enable more consistent product and cell concentration. So in a continuous operation, you typically have cell bleeding in order to maintain a certain cell concentration to achieve steady state. So initially, this could be done in a very manual way where you have manual control, take offline samples, and then do a batch removal of the cell bleed to accomplish that steady cell concentration higher. But really, by automating that and using a closed-loop probe, you can really smooth out that steady state and get a steady-state operation. And again, much better cell concentration and more consistent. Cost of media can be something to consider in the cost of goods analysis for perfusion and continuous. Here's just an example of a well-defined medium which contains LongR3. LongR3 is a cell culture growth factor supplement that's 200 times more potent than insulin. So what this graph is essentially showing is that the cost of LongR3 addition is 9% more, but in fact with adapted cells you can accomplish almost 100% increase in production per liter. So the economic benefits can be vastly improved. Right. Perfusion media, although your volumes increases, typically of the media could be leaner, and then with the right ingredients. So to finalize, manufacturing drivers for change. So continuous processing reduces manufacturing risk in a number of ways. The capital investment and technology transfer through reduced COGS. lends itself to single-use facilities and, of course, can be applied to fed batch facilities as well through cell banking and N-1. But what we see is a move towards continuous processing is a paradigm shift. And with these new technologies and the changing regulations, that transition much easier, we're seeing more approved processes in the standard, not only for continuous processing, but also for fed batch processing. That would be my final slide. Open up for any questions.
Thank you,